Hello students, the topic of today's class is membrane lipids. Now let's look at the main objectives of today's module. Number 1, to study about the different important lipid components of cellular membranes and their properties. And secondly, to study how the composition of these different types of lipids affect the physical properties and function of cellular membranes. Biomembranes are large, flexible sheets with a bilayer structure that serve as a boundary of cells and their intracellular organelles. Lipids contribute to the structures of a variety of membranes in different parts of cells and in different organisms. The composition of these lipids varies considerably depending on the type and function of the membrane. The percentage of lipids and protein in different membranes can range from 80% lipid in myelin to only about 25% in mitochondria. A typical biomembrane contains 3 classes of lipids: phosphoglycerides, sphingolipids, and sterols, which differ in their chemical structures, abundance, and function in the membrane. Almost all membrane lipids are amphipathic molecules, such as phospholipids or glycolipids, having both hydrophilic and hydrophobic parts. Neutral lipids, such as triacylglycerols, have little part in membrane structure. Among the membrane lipids, all phosphoglycerides and certain sphingolipids are phospholipids. Let's study the phosphoglycerides first. Phospholipids are abundant in all biological membranes. Phospholipids, together with membrane proteins and cholesterol, are involved in many cell functions, such as in the control of cell shape, compartmentalization, the storage of compounds, ion transport, metabolism, cell signaling processes, and cell fusion processes. A phospholipid molecule is constructed from 4 components: fatty acids, a platform to which the fatty acids are attached, a phosphate, and an alcohol attached to the phosphate. The fatty acid components provide a hydrophobic barrier, whereas the remainder of the molecule has hydrophilic properties to enable interaction with the environment. In phosphoglycerides, the hydroxyl group at C1 and C2 of glycerol are esterified to the carboxyl groups of the 2 fatty acid chains. The C3 hydroxyl group of the glycerol backbone is esterified to phosphoric acid. The major phosphoglycerides are formed due to the formation of an ester bond between the phosphate group and the hydroxyl group of one of several alcohols like the amino acid serine, ethanolamine, choline, and inositol, and their corresponding names are phosphatidylserine, phosphatidylethanolamine, phosphatidylcholine, and phosphatidylinositol. Phosphatidylcholines are the most abundant phospholipids in the plasma membrane. Let's study the sphingolipids. A second class of membrane lipids is sphingolipids. These lipids, based on a sphingosine rather than a glycerol backbone, are also widespread in membranes but are particularly abundant in nervous tissue. Generally, they tend to be concentrated in the outer leaflet of the plasma membrane. They are derived from sphingosine, an amine alcohol with a long hydrocarbon chain, and contain a long-chain fatty acid attached to the sphingosine amino group. Like phosphoglycerides, some sphingolipids have a phosphate-based polar head group. Sphingomyelin is the most abundant sphingolipid. Other sphingolipids are amphipathic glycolipids, whose polar head groups are sugars, that are not linked via a phosphate group, and so technically they are not phospholipids. Glucosyl cerebroside, the simplest glycosphingolipid, contains a single glucose unit attached to sphingosine. In the complex glycosphingolipids, called cerebrosides, one or two branched sugar chains containing sialic acid groups are attached to sphingosine. Glycolipids constitute 2 to 10% of the total lipids of cell membranes. They are most abundant in nervous tissue. Sterols. Cholesterol and its analogues constitute the third important class of membrane lipids, the sterols. The basic structure of sterols is a 4-ringed isoprenoid-based hydrocarbon. Cholesterol has a hydroxyl substituent on one ring. Although it is most entirely hydrocarbon in its composition, it is amphipathic because its hydroxyl group can interact with water. Because it lacks a phosphate-based head group, it is not a phospholipid. Cholesterol is especially abundant in the plasma membranes of mammalian cells, but is absent from most prokaryotic and all plant cells. The structures of the principal yeast sterols, ergosterol, and plant phytosterol, like stigmasterol, differ slightly from that of cholesterol. The small differences in the biosynthetic pathways and structures of animal sterols are the basis of most antifungal drugs currently in use. As much as 30 to 50% of the lipids in plant plasma membranes consist of certain steroids unique to plants. Between 50 to 90% of the cholesterol in most mammalian cells is in the plasma membrane and associated vesicles. Cholesterol and other sterols are too hydrophobic to form a bilayer. Instead, they intercalate between the phospholipid molecules to be incorporated into biomembranes and thus prevent too close a packing of the phospholipid molecules. This helps in maintaining membrane fluidity and at the same time providing the necessary rigidity required for mechanical support. Let's look at the properties. Lipid bilayers are highly impermeable to ions and most polar molecules. Lipid bilayer membranes have a very low permeability for ions and most polar molecules. Water is an exception since it traverses such membranes relatively easily because of its small size, high concentration, and lack of complete charge. However, sodium and potassium ions can cross these membranes 10 to the power 9 times as slowly as does water molecules. The permeability of small molecules is correlated with their solubility in a non-polar solvent relative to their solubility in water. An ion such as sodium ion traverses membranes very slowly because the replacement of its coordination shell of polar water molecules by non-polar interactions with the membrane interior is highly unfavorable energetically. Movement of lipids in biomembranes. In the two-dimensional plane of a bilayer, thermal motion permits lipid molecules to rotate freely around their long axis and to diffuse laterally within each other. Because such movements are lateral or rotational, the fatty acyl chains remain in the hydrophobic interior of the bilayer. At 37 degrees Celsius, a typical lipid molecule exchanges places with its neighbors in a leaflet about 10 to the power 7 times per second and diffuses laterally several micrometers per second. These diffusion rates indicate that the lipid bilayer is about 100 times more viscous than water. When artificial pure phospholipid membranes are cooled below 37 degrees Celsius, the lipids can undergo a phase transition from a liquid-like state to a gel-like state. Below the phase transition temperature, the diffusion of lipids drops sharply. At normal physiologic temperature, the hydrophobic interior of natural membranes has a low viscosity and fluid-like consistency. In pure membrane bilayers, phospholipids and sphingolipids rotate and move laterally, but they do not spontaneously migrate or flip-flop from one leaflet to another. This is because the energetic barrier is too high and migration would require moving the polar head group from its aqueous environment through the hydrocarbon core of the bilayer to the aqueous environment on the other side of the membrane. Special membrane proteins are required to flip membrane lipids and other polar molecules from one leaflet to the other. Effect of lipid composition on the physical properties of membranes. A typical cell contains many different types of membranes, each with unique properties derived from its particular mix of lipids and proteins. Several phenomena contribute to these differences. For example, the relative abundances of phosphoglycerides and sphingolipids differ between membranes in the endoplasmic reticulum, where phospholipids are synthesized, and the Golgi apparatus, where sphingolipids are synthesized. Sphingomyelin percentage is about 6 times as high in Golgi membranes as it is in the endoplasmic reticulum membranes. In responding to differing environments throughout an organism, different types of cells generate membranes with differing lipid compositions. The degree of membrane fluidity depends on the lipid composition, the structure of the phospholipid hydrophobic tails, and temperature. Van der Waals interactions and the hydrophobic effect cause the non-polar tails of the phospholipids to aggregate. So, long saturated fatty acyl chains have the greatest tendency to aggregate, packing tightly into a gel-like state. Phospholipids with short fatty acyl chains have fewer van der Waals interactions and they form more fluid bilayers. Likewise, the kinks in cis-unsaturated fatty acyl chains form less stable van der Waals interactions with other lipids and hence there are more fluid bilayers. Straight saturated chains, on the other hand, can pack more tightly together. The cholesterol is important in maintaining the appropriate fluidity of natural membranes, a property which is essential for normal cell growth and reproduction. Cholesterol restricts the random movement of phospholipid head groups at the outer surfaces of the leaflets. Interaction with long hydrophobic tails of the phospholipids immobilize these lipids and thus decrease membrane fluidity. This property helps to organize the plasma membrane into discrete subdomains of unique lipid and protein composition. Lipid composition also influences membrane thickness and curvature. Membrane lipids are modified to maintain fluidity at low temperatures. Poikilotherms, organisms unable to regulate their own temperatures, which grow at low temperatures, have a pattern of membrane lipids possessing lower phase transition temperature than those of the same organisms existing under hotter conditions. Furthermore, following changes in environmental temperature, an alteration in the composition of membrane lipids is seen. Although an increase in fatty acyl unsaturation at lower temperature is best known, it is not the only change that can take place. The changes in membrane composition caused by lower growth temperature in different poikilotherms are retailoring of molecular species, like a quick change involving swapping acyl chains on the glycerol backbone, increase of fatty acyl unsaturation, shortening of fatty acyl chains, alteration of fatty acid type, alterations in lipid classes like phospholipid interconversion from phospholinositol to phosphatidylcholine, change in lipid to protein ratio of the membrane. Now let's look at the asymmetric distribution of membrane lipids. A characteristic of all biomembranes is an asymmetry in lipid composition across the bilayer. That is, lipid composition is different in the exoplasmic and the cytosolic leaflets. Although most phospholipids are present on both membrane leaflets, some are commonly more abundant in one or the other. Almost all phosphatidylcholine and sphingomyelin, which form less fluid membranes, are found on the exoplasmic leaflet, while phosphatidylethanolamine, phosphatidylserine, and phosphatidylinositol, which form more fluid membranes, are present preferentially on the cytosolic leaflet. Glycolipids are present exclusively on the extracellular leaflet. The preferential location of membrane lipids on one face of the bilayer is necessary for a variety of membrane-based functions. For example, the head groups of all phosphorylated phosphatidylinositol, an important source of second messengers, face the cytosol. Stimulation of many cell surface receptors by their corresponding ligand results in activation of the cytosolic enzyme phospholipase C, which can hydrolyze the bond connecting the phosphoinositols to the diacylglycerols. Phosphatidylserine also is normally most abundant in the cytosolic phase of the plasma membrane. In the initial stages of platelet stimulation by serum, phosphatidylserine is briefly translocated to the exoplasmic phase by a flippase enzyme, where it activates enzymes that participate in blood clotting. When cells die, lipid asymmetry is no longer maintained, and phosphatidylserine is increasingly found in the exoplasmic phase of the plasma membrane, which is indicative of a cell undergoing apoptosis or programmed cell death. So let's summarize the points that we have covered so far. Number 1, the phospholipid bilayer, the basic structural unit of all biomembranes, is a two-dimensional lipid sheet with hydrophilic faces or leaflets and a hydrophobic core. Secondly, the primary lipid components of biomembranes are phosphoglycerides, sphingolipids, and sterols, which differ in chemical structure, abundance, and functions in the membrane. Thirdly, most lipids are laterally mobile in the bilayer. Membranes can undergo phase transition from fluid to gel-like states depending on the temperature and lipid composition. Fourthly, lipid composition influences the physical properties and functions of the plasma membranes. Fifthly, membrane lipids modification and changes in membrane composition take place in poikilotherms to maintain membrane fluidity even at cold temperatures. And lastly, membrane lipids not only affect membrane shape and function, but also help to anchor proteins to membranes, modify membrane protein activities, and transduce signals to the cytoplasm.